Good afternoon, everyone. Today, I want to talk about two statistically distinct patterns from statistically distinct patterns based on two different properties. One is based on the molecular structure, which is captured by chirality, and the other one is captured by hierarchical biochemical organizations. And in the end of the talk, I would like to discuss how these two patterns related to each other and give us how these laws of chemistry and principle of biology is related to each other. So before and after the LUCA appeared, the systems before the LUCA appeared, the systems are more dominated by laws of physics. But after that, it is usually considered more dominated by, organized by principles of biology. But yet, we don't know what exactly happened between these two processes. But we have some general, even though we don't know the rigorous theory between these two, still we have some general, we have idea about the general relationships. So usually the principle of biology is considered as constraint for the laws of chemistry, and the laws of chemistry, the chemical space is governing the space of the chemical molecules and their complicated collective relationship emerged to have some organization which follows principles of biology. And in our study, we are interested in the universal scaling behavior across different levels of biological system as constraint. And for emergent property, we are focused on the Homochirality, and which is emerged from, which we consider emerging property from complex collective interactions of chirality, molecular chiralities. So to do so, we adopted network theory and the biochemical networks, which is independent of the very details of the systems and the components. In the seminal paper written by Zhang et al., they studied biochemical networks by annotating the enzymes and used the KEGG database, and they built the metabolic network. And these networks even can be further abstracted to connections between the biochemical compounds, and they are connected to each other when they participated in same biochemical reactions. And what Zhang et al. Found is that if the connectivity patterns follows power law degree distribution, can be written by this formula, and then this power law exponent is power law exponent lies between certain range for biological systems. So many people were asking if this is characteristics of biology. The answer, unfortunately, the answer is no. First of all, there are lots of social networks or technology networks which shows the same property, and secondly, they used only 46 biochemical networks, which is too small to decide to extract any general principles. And then they only focused on the individual organism level of biochemical networks. And as you know, the biochemical living process is really difficult divide or isolated. And these days, people are more interested in the finding the understanding planetary scale organization of life. So this is network visualization. These nodes are chemical compounds, and they are connected to each other when they share the biochemical reactions. And these size of nodes indicates the number of connections and number of reactions they are participating. And this consists of over 8,000 biochemical reactions and 9,000 biochemical reactions and over 7,000 biochemical compounds. And so we consider the three different levels of organizations. First, individual organism level, and then ecosystem, and then the whole biosphere. And these are embedded hierarchical structure, as you can see, and using the KEGG data and the JGI Patrick database, we generated three different levels of biochemical networks. For individual, we had over 20,000 genomes, and then to generate ecosystem network, we utilized over 60,000 metagenome data. And as a proxy of a biosphere, we utilized every catalyte, every reactions in the KEGG data. And the first result we found is that we measured well-established network measures for individual networks and over 6,000 ecosystem networks. And we found that there are these two scaling patterns follow same functional forms, even though they are quantitatively different, but there are qualitative organizing principles exhibit from both levels of organizations. And then we checked if we tested if this is a result of random chemistry or it's originated from biological organization principle. So to do so, we generated 5,000 random reaction network. The reaction itself is from KEGG data, but the combinations are randomly those combinations are randomly connected. And as you can see that these random reaction networks doesn't share the same form of scaling laws. And then we could see that these biochemical networks, these statistically distinct patterns actually explains it's originated from biological organization principles. So next, our next focus was about the chiral nature of these biochemical networks. We used the same system and in the different levels of organization. And chirality can be simply defined by the object whose mirror image cannot be imposed exactly. And historically, the study was focused on the macromolecules like DNA, RNA, or proteins. However, since we consider the function of life as emergent phenomena, we were considering homochirality. We were thinking homochirality is also probably emergent property from messy chemical systems. To test this hypothesis, we annotated the whole biochemical networks with the biochemical molecules of the whole biosphere as chiral molecules or achiral molecules. Here, the blue color indicates the chiral molecules, and the pink nodes indicate achiral molecules. And you can see that actually these are not really, you cannot really separate the two groups, and there are very complicated relationships between them. And I tested the scaling patterns for characterizing this chiral nature of biochemical networks at different levels. So these as you can see that the ecosystem and the bacteria, eukarya, archaea, which represented, these three groups represent the individual organism level biochemical networks, and these are decreasing as the total number of compounds in the system increases, and this is a biosphere. And on contradicting those patterns, if we look at the random networks, the chiral compound percentage is really independent of the size of networks. So we can see that there is distinct statistical patterns which can differentiate the biochemical networks from random networks in terms of the molecular chirality. So since this distinct pattern is observed, we were interested in how we can explain in terms of evolution of biochemical networks. So we used the network expansion algorithm, which starts with a random certain set of seed compounds, and when this can activate some biochemical reactions, and then we include those reactions and their products. And we repeat this process as the time step increases. So as you can see that this time evolution pattern really dependent on two factors. One is the selection of seed compounds. The other one is the structure of the background network. Since we have, for the background network, we adopted the whole KEGG network, and then for the first step, we selected 6 achiral primordial seed compounds, and this is the result. The x-axis indicates time steps. Y-axis indicates the number of nodes newly added to the expansion. And you can see that in the beginning, and so yellow indicates the old compound, number of all new compounds, and the blue indicates the number of new chiral compounds entered the expanded network. As you can see that in the beginning, the number of new chiral compounds really grows slowly, and it is more dominated by achiral compounds. But then there is some peak. After a few peaks, chiral compounds really almost three times more than achiral compounds, because this gap is equivalent to number of achiral compounds. And then later, when all the new, there are no more new compounds entered and expanded the network, it's more like the network all includes just chiral molecules. So this is only first step, but it gives some idea, it gives a solid idea about how the achiral prebiotic precursor might utilize this web of biochemical reactions to have a certain, to have the structure like the modern biology. Sorry. And then there are a few statistical results that we found based on the chirality. And this is conversion rate between chiral and achiral compounds through biochemical reactions. So the y-axis shows the chiral compounds rate percentage in reactants, input of the biochemical reaction, a biochemical reaction, and the x-axis indicates chiral compounds in products, or products biochemical compounds. And as you can see that 3 points has most frequently appeared and in this diagonal, but also there are some high frequently appeared cells here. So here you can see that when even though reactants are 100% chiral, they are converted to some achiral molecules through biochemical reactions. Also, this shows the relationship amongst number of chiral centers and the molecular weight. It is usually expected that these will be just a linear relationship, but it is a linear relationship, but you can see that there are 2 distinct groups that follows different increasing rates, and these are the same relationships, but in different domains of life. It's a conclusion and acknowledgement. Thank you. Any